Welcome everyone for the fractional calculus seminar series. Today we have Professor Sergei Fedotov, who obtained his PhD from Uriel Federal University. Professor Fedotov has held research positions in London in 1990, from Aachen from 1993 to 1995, Wuppertal in 1997, Berlin 1998, before joining the Department of Mathematics at UMISC in Manchester as a lecturer in 1998. Since 2005, he is a professor of applied mathematics at the Department of Mathematics, University of Manchester. Professor Fedatov's research focus on random walk theory and reaction transport system. He has successfully applied anomalous random walk ideas to a broad range of analytical studies related to non-Markovian transport phenomena in physics, chemistry, and biology. It's very nice to have you here, Professor Fedatov. You may please begin your talk.
Thank you very much, Pavan, for invitation to give a talk. It's a pleasure. Also, thanks, Arran, for inviting me to give a talk. Today I'm going to talk about fractional PDEs, in which anomalous exponent, not a constant, but a function of space coordinate. So, of course, I should mention a recent nice review by Roberta Garapa, Ginzi, and Francesca Mainardi about variable order fractional equations, but then anomalous exponent's function of time. Today is a function of space. So mostly I focus on derivation of this equation rather than solutions. And of course, I've got the reason why, because there is no unique variable order fractional PDE. Depending on underlying random walk, we could derive different mesoscopic fractional equations, and those equations has completely different solutions. So it's a high sensitivity on the modeling process. So there is no unique variable order fractional PDEs. And of course, I will give you examples and try to illustrate in terms of examples from physics and biology. But first, let me give a brief outline of my presentation today. At the beginning, of course, I will introduce variable order time fractional diffusion equations. Of course, some history, who first derived these equations. And then I turn to particular problems of subdiffusive transport in heterogeneous biofilms. The reason why I'm going to discuss this particular example from biology is that it's a motivation to derive so-called variable order reaction-subdiffusion equation. Because in heterogeneous biofilms, everything depends on space coordinate, diffusion coefficient, drift, anomalous exponent, etc. So, and of course, we will derive equation and discuss the property of this equation. So this is my plan. Okay, let me start with the classical time fractional Fokker-Planck equations. Of course, this equation describes evolution of arbitrary distributions towards some stationary distribution. It might be like Boltzmann distribution, Boltzmann distribution and other distribution. And the main difference between this equation and the classical Fokker-Planck equation is just the rate at which convergence happens. So because of, on the left-hand side, you can see Caputo derivative, this convergence is very slow. The reason why I mentioned this convergence towards stationary distribution, it will be clear in a few minutes. Now, let me mention that this equation involves three parameters. So on the right-hand side, we can see drift, also diffusion, and of course, anomalous exponent alpha, which varies from 0 to 1. It's a lot of literature on that equation, of course, in physics and mathematics. Of course, I should mention a very nice review by Ralph Metzler and Yossi Klafter, 2000, in Physics Reports. Also, many people contributed to this equation, like Eli Barkai and many others. Very nice book by Sokolov. So this equation is well studied. Usually, in physics literature, this equation written in terms of Riemann-Liouville derivative. In mathematical literature, usually it's written in terms of Kaput derivative. And of course, there is a reason. I'm not going to mention all mathematical work, but I should mention very nice work by Nikolai Leonenko, Mark Meerschaert, and Alla Sikorsky, in which they study time fractional Pearson diffusion when drift and diffusion coefficients are polynomial functions, first and second degree. So they applied a variety of technique, eigenfunction expansion, of course, times change using inverse stable subordinator. And they showed how we have a convergence of arbitrary distributions towards some stationary distribution. For example, if V drift is linear and diffusion constant, of course, we have convergence to Boltzmann distribution. And the interesting example, for example, in that paper, Cox-Ingersoll-Ross process, in which drift is linear function, and the diffusion coefficient is also a linear function. And then for positive value of x, we have convergence toward gamma distribution. Now, very natural question arises. Why we have two parameters, drift and diffusion are space dependent, but alpha is constant. Of course, there is mathematical reasons. It's easier to treat because if you look at this equation on the right-hand side, we have a very nice operator. On the left-hand side, we have a Kaput derivative. We can apply separation of variables. But if we remember the reason why we have these parameters, then it's clear that alpha might be also space dependent. Because like drift, why drift is space dependent? Of course, it's because of external force might be dependent on x. In heterogeneous medium, diffusion also not constant. It's space dependent. But what is alpha? Sorry, let me remind you. Of course, alpha is a measure of strength of trapping. So the reason why we have Kaput derivative on left-hand side, if we look at underlying random process, then the physical reason is that it's a long trapping. So waiting time or residence time distribution, it has a heavy tail distribution. And if the first moment is divergent, then we have that fractional equation. We know that. And alpha gives the strength of this trapping, because alpha gives us the rate at which this power law function decay with time. So alpha depends on x. It might be the case. So, and that question, of course, asked by many people a long time ago. And in 2005, three people, Churchkin, Gorenflo, and Sokolov, first time they derived from continuous time random walk, very simple symmetrical random walk, they derived the following equation. So now you can see that they derive in terms of Riemann-Liouville derivative, not Kaput derivative. And because this operator involving anomalous exponent, and now let me tell you that for the rest of this talk, I'm going to use for anomalous exponent notation mu. So please remember at least for 1 hour that anomalous exponent, not alpha, but mu. So this anomalous exponent mu depends on x, and this operator inside second derivative. Well, of course, it involves diffusion coefficient, which is also depends on x via dependence of anomalous exponent. And then anomalous exponent, of course, varies from 0 to 1. it's difficult to solve that equation actually, and because the standard method doesn't work. And for that reason, Churchkin, Gorenflot, Sokolov split the whole space in two subspaces and then separately solve equations for constant mu and then organize properly boundary condition. And they found interesting solutions. Also, I should mention, of course, Nikolai Korabel, Eli Barkai work in 2010. They also analyzed that equation and found very interesting properties. Later, me and my PhD students, Daniel, also discussed that problem, and I'm going to discuss it anyway. Also, I should mention mathematical work in that area. Mladen Saov and Bruno Tualdo wrote a very sophisticated paper in which they treated the case when anomalous exponent space dependent. Now, let me discuss unusual property of this equation. It's easy to derive also extension of that equation involving advection term. So as long as you consider non-symmetrical random walk, you can easily to find that you have a drift term involving this operator with anomalous exponent, mu. A long time ago, 2012, together with my PhD students, we made very simple experiment. So we consider that equation on infinite domain and ask ourselves, what happened if anomalous exponent mu is not just constant, but constant plus something, a small variation, very small. So if you look at the picture, so we have a constant mu everywhere, apart from this little zone. And we made experiments, numerical experiments, because it's difficult to solve that equation. And we found that this small variation leads us a drastic change in asymptotic behavior of probability density function for large time. And we found that Gibbs-Boltzmann distribution is no longer stationary solutions. And it was quite disturbing. So historically, I remember, you see, if you make a small perturbation of drift or diffusion coefficient, then it does not impact on a stationary distribution too much. It's just small variation. But when we have small variation in anomalous exponent, it's a huge impact. So let me explain what kind of impact we have. So this is experiment. We have a finite domain with 100 boxes. And first we, of course, solve Fokker-Planck equations for the case when everywhere anomalous exponent 0.5 for all boxes. And we found that's just Gibbs-Boltzmann distribution. We organized, of course, the drift term such that we have this nice distribution. So we have a very nice convergence. But then what happened, we say, okay, anomalous exponent everywhere 0.5, apart from one box. In this particular case, it's a box 42, in which anomalous exponent 0.3. We ran experiments and we found that all particles concentrated in that box. So there is no convergence whatsoever toward nice stationary distribution, and particles started to concentrate on that particular point. So it was very disturbing because it looks like the fractional equation with space-dependent anomalous exponent, it's not structurally stable. And it means it's not good for modeling because a small variation of mole parameter drastically changed the solution. But at the same time, we thought probably it's an advantage of that equation because it describes a very interesting phenomenon, and we call it anomalous aggregation. So, and of course, it's physically easy to explain. So at the point 42, we have the trap And the strength of this trap, 0.3. And of course, that trap attract all points. It's actually very slowly. So then we started to do experiment, and then we make experiment further. Oh my God. Right. Okay, I'll explain later. So now in 2019, we decided to find the solutions of that equation for the simple case when anomalous exponent is a linear function. But first, we decided to derive equations, and we come up with different equations. So when we use local trap model, the equation was, of course, the same as in Churchkin-Gorenflor Sokolov paper. But when we use a barrier model, and I explain this model in a few minutes, then we have this equation. So, and it's clear a big difference between these two equations, because here you can see this derivative with respect to x appears here, but here we have like a second derivative. So, right, okay. And these two equations actually have got completely different asymptotic solutions. So, for example, if for barrier model we start with arbitrary distribution in finite domain, then sooner or later we have uniform distribution because of this term. So uniform distribution is a standard, of course, distribution for this kind of equation. But for local trap model, we have anomalous aggregation. Now the question is, which model is better for subdiffusive moment in heterogeneous environment? This is not trivial question, because in practice, when we derive something like in physics, we expect that, for example, Fokker-Planck equation has equilibrium solution, like Boltzmann distribution. But subdiffusive system, they are not at equilibrium. So they describe non-equilibrium systems, and this criteria that at the end we will have equilibrium distribution is not the case. So how to check which equation is right? This is really a serious problem. And now let me explain how to derive these two models. So, of course, if you formulate continuous time random walk model, then very simple symmetrical random walk model, then you will end up with a master equation for trap model. So if you look at this equation, clearly the rate at which probability density function change with time depends on the flux out of the trap. And we have two terms of particle coming to this trap and the escape rate involving Riemann-Duval derivative. But for barrier model, we have to formulate the problem slightly different. We just assume that between boxes, we have a barrier, and the rate at which probability density or mass probability varies with time depends now on four terms. You can see this is a rate of particles coming left, a rate of particle coming right, and of course we have two rates, particle coming to this trap model. But now look at this. This is very important case. You see the escape rate on the right, like plus, depends on mu, but the value of mu evaluated at the point x plus a over 2. So like at barrier. So if we evaluate anomalous exponent at the point x, then we have trap model. And if you evaluate x plus a over 2, then we have barrier model. At first glance, it looks not big difference because at the end we expand in terms of a. But this small difference actually create a huge implication, and let me explain this implication. So if we reformulate variable order fractional PDE in terms of the flux, so this is the standard equations, and now look at the flux of the particle. So for barrier model, we have almost like Fick law. The flux is proportional minus the effective diffusion coefficient, anomalous one. We have, of course, the Riemann-Liouville operator, and proportional to the gradient of P. This is quite natural. But for local trap model, we have the same term, of course, but we have extra drift. And this drift proportional to the derivative of anomalous exponent with respect to x. So, of course, when mu is constant, this term disappears, but it looks like chemotaxis. So as long as we have different value of mu in different spaces, this effect induces that kind of drift in the movement of particles. And also interesting things, we did calculations and found that this drift involves interesting operator. Look at this operator. It's almost like Riemann-Liouville operator, but the difference is that here in denominator we have a slowing varying function of time. So apart from decay due to Power law, we have an increasing but slowly increasing function of t. So the decay of total kernel is very, very slow. And from a mathematical point of view, this term actually induces anomalous aggregation. So and we found a solution when time tends to infinity. Then asymptotic solution of variable order fractional PDE looks like this. So this is published in 2019. So it's easy to illustrate this. This is the visualization of that solution. We start from uniform distribution. And of course, if we start from uniform distribution for barrier model, it will stay uniformly distributed. But for trap model, we have this funny drift, and this drift push particle from the right to the left, and it's very, very slowly. But then we had kind of this distribution, and asymptotically, it tends to delta function at the point 0, but the rate is very, very slow. So that's the huge difference between local trap model and barrier model. So we also make experiment with drift. This is a standard drift, and the anomalous drift is much stronger than the standard drift, and also we have the same solution in long time limit. But we also have very interesting observation. So if we start, let's say, from this step function at the beginning, and then run the problem, then in intermediate time, let's say t equal to 5, we have almost like Boltzmann distribution. You see? But then this Boltzmann distribution completely destroyed. So what I'm trying to say that in the intermediate asymptotics, this Boltzmann distribution is fine. But of course, in long time limit, we have this kind of distribution. Right, that's a problem with variable order fractional Fokker-Planck equation. And because at that time we analyzed the movement of lysosomes inside cells, my PhD students Daniel Hunt made experiments, and we found that it's actually well known that lysosomes inside cell, they move subdiffusively. This is a famous paper, Cell Reports 2018. And but this is our picture. Daniel made it in a lab that you see this is green particles is lysosomes. They concentrated and got large cluster around spinnicular nuclear region. And we tried to fit our solutions with the data, and we have a very good fitting. So that paper published in 2021. So it seems we can confirm that anomalous aggregation in terms of real picture. But again, the question remains, what is the right equation? Barrier model or a local trapping model? And let me repeat that local trapping model is derived by Chochkin, Gorinfo, and Sokolov. But also you can derive different equations. Just tiny change in underlying microscopic random walk model. Right, but now I'm going to talk about biofilms. The reason why I'm going to talk biofilms because it gives us an example in which we have a real system, in which we have a heterogeneous environment. It means that diffusion, anomalous exponent are space dependent. But first, let me remind you what is biofilm. Actually, if you look at this picture, biofilms is a community of microorganisms. You can see different bacteria. And these bacteria, they stick to each other, and they stick also on the particular surface. And then all of them embedded in a so-called extracellular polymeric substance. So it's like very sticky. It involves many things like mostly biopolymers and like the DNA, lipids, proteins, name it. So, and in experiment, people showed that it's a very slow penetration of antibiotics. So if we want to treat this disease involving biofilms, it's extremely difficult because if you look at the picture, then antibiotics, yellow color, may fail penetrate beyond some surface area in biofilms. And usually that problem is treated in terms of classical reaction diffusion equation. So you can see that if we introduce the density of antibiotics rho, it involves, of course, diffusion depends on X. So X actually along the depths of biofilms. Real biofilms really heterogeneous because of different bacteria of different phenotypes. And this is really X dependent. It's a seriously heterogeneous system. So you have to take into account diffusion coefficients depend on X. Also, a reaction usually depends on X, also might depend on the density. So people analyze the problem of poor penetration of antibiotics in terms of classical reaction diffusion equation. But recent experiments this is like one slide about the importance of that problems because it's like a very scary picture, like almost 5 million deaths associated with drug resistance bacterial infection. Basically, it's written here, the biofilms is just a barrier, penetration barrier for drug molecules. And of course, it enhances bacterial resistance to antibiotics. And now we think in our group that biofilms can be treated or tackled in terms of reaction-subdiffusion system involving time fractional PD. And already I mentioned it because it's highly heterogeneous. So now there's now overwhelming evidence that transfer of antibiotics is subdiffusive. Many papers already published. I should mention these two. And also I need to mention two papers by Ralph Metzler together with co-author. They analyzed reaction-subdiffusion equation for biofilms. But no surprise, they use anomalous exponent as a constant. But in real biofilms, the anomalous exponent, of course, is not constant, is depth dependent. And again, it's a lot of evidence for that. So now let me explain the mathematical challenge we face when we try to analyze slow penetration of antibiotics. First of all, it's a transportive subdiffusive. Then we need to implement nonlinear reaction term and degradation terms. Usually it's depth dependent and also might depend on the density of antibiotics. Also, we have, of course, depth dependent anomalous exponent and anomalous diffusion coefficient. And you see, the reason why I discussed passive case, that everything depends on underlying random walk, microscopic picture. So mesoscopic fractional equations, the form of them depends on these microscopic assumptions. And now here, together with reactions, the situation becomes very, very complicated. And we cannot rely also on phenomenological argument. So we can't simply write down equations like this. And that's what I'm going to explain. So here we have transport, of course, heterogeneous. Also we have reaction, but reaction and transport is separate. But for reaction diffusion or reaction subdiffusion, it is not. So let me remind you some ideas about this. If we have classical advection diffusion PDE, like this one, if we want to take into account reaction, we just put on the right-hand side nonlinear terms like this, where theta is a nonlinear degradation rate. So rho is just density of particles. So it might be nanoparticles, it might be large antibiotics molecules. But if we consider time fractional PDE with anomalous transport, that's also already well known that we can't simply put on the right-hand side nonlinear terms. This doesn't work. And it's not a good model for subdiffusion with degradation reaction. Actually, many people in the last almost 20 years studied that problem, how to derive reaction subdiffusion equation. And again, I should mention several people involved in this research. Of course, I should start with Igor Sokolov, who made a pioneering work in this area. He first derived reaction subdiffusion equation. At the same time, also a group in Australia, Bruce Henry, Langlands, Chris Angstman, they also produced very nice work in that area. Shkilov from Kiev, many people, Werner Horst Hemke, my co-author. Also, I need to mention, of course, Spanish researchers like Juste, Abad, Vicente Mendez with Daniel Campos. There's many work. Some mathematical work like Nek and Nepomniachtchi from Israel. There's many people. I can't mention all of them. And they derived a variety of reaction subdiffusion equation. But of course, most of the time, 99%, anomalous exponent is constant. So now the challenge is to implement a reaction term together with anomalous exponent, which is space dependent. I will just mention one previous work. It's in 2006. So if we have simple problem, reaction diffusion equation, we have transport, diffusion minus degradation. So it has a very nice solution. It's not a problem to deal with that equation in different boundary conditions. But if we start with continuous time random walk, derive master equation with reaction, and then consider diffusion approximation, we end up with this equation. So you see, theta is a constant, but it's very interesting phenomenon happens. Look at right-hand side of this equation. So here, of course, we have degradation, linear degradation, but it turns out this degradation rate also appears in transport operator. So transport and reaction is not separate anymore. And also it's very interesting observation that this reaction rate plays the role of tempering, which has a huge implication on the solution of this time fractional reaction subdiffusion equation. So the main idea is that if you start with microscopic model, then you might end up with a different reaction-subduffusion PDE with different properties. And that's what I'm going to talk today. And again, we need this equation not just because we want to derive something, because this problem with poor penetration of antibiotics, that problem needs our new equations, and that they are not yet available. And I explain why they are not available in a minute. Okay, now I would like to discuss, I still have 20 minutes, I would like to discuss a structural density approach. The reason why I want to discuss it, because we want to implement reaction term. We know that continuous time random walk model usually formulated in terms of waiting time and jump distribution, and usually we write down renewal equations, integral equations, and it's not easy to implement reactions when we have these integral equations. And in my opinion, the best way to implement something into continuous time random walk, it might be reaction, it might be different sort of non-linearities, if we start with structural probability density, which is a probability of finding particle at a position. So this is lattice at the time t, but with the h tau since the last jump. So h tau means not total h from the beginning, but since the last jump. And this is a standard method in probability theory, but it's very useful because we formulate problem as Markovian. And as long as the problem is Markovian, it's easy to implement anything. For example, if we have local trap model, of course, now everything specified not in terms of waiting time or residence time distribution, but in terms of rate. And rate now depends on residence time and space, of course. So gamma, if it's a decreasing function of tau, and then we could, of course, derive a classical power law distribution. So the first term on the right-hand side describe all non-Markovian phenomenon when we reduce the system, when we eliminate tau. It's a very useful approach, but look at this term. Also, this explains degradation terms. And so, of course, this is not conserved probability anymore. For barrier model, we have to take into account that we have to calculate escape rate, not in terms of x. We need to shift it at x minus a over 2. And we have two barriers at this point and at this point. So also we have reaction term. In this structural density approach, we have to also specify boundary conditions at zero waiting time. Well, this is quite natural. It's a symmetrical random walk. And so we just integrate over tau. So as I mentioned, the depth and age-dependent escape rate gamma allows us to capture all non-Markovian effects on mesoscopic level. If we, let's say, assume that residence times has survival probability in Mehta-Flair function, then of course we can evaluate this gamma by using standard method. So yes, so our aim, of course, to obtain various coarse-grained variable order reaction-subdiffusion fractional PDE for this mass probability mass function, if it's on lattice. We just integrate this structural density with respect to tau. And if we do that and do calculations, of course, I skip all details, then you will see that for local trap model, we have escape rate, looks like this. And you can see for space-dependent degradation rate, it appears also as a product. So again, it looks like a tempering fractional derivative. But here we use tempering not just artificially to cut long waiting time, it happens quite naturally. So degradation terms actually plays a very important role because it kills those particles that stay a long time in trapping. And the implication I will discuss later. For barrier model, we have to evaluate of course, at barrier, and we can come up with different equations. So also we can formulate master equation for local trap model and barrier model. What I have to tell you that for local trap model, we can derive equations quite easily, but for barrier model, it's not easy to derive diffusion approximation. You can come up with different models, and now I'm not going to discuss it because this is just work in progress. But I need just to mention that for local trap model, we have equation. So again, let me repeat. Now we need to obtain nonlinear fractional reaction-subdiffusion equation with anomalous exponent, which depth dependent, because that's important for this particular model of biofilms, and nonlinear reaction rate, which is also degradation rate. And the governing equation for the mean density of drug molecules. So you see, when I discuss this model, this is a linear model for probability mass function. But also we can formulate the similar equation for the mean field, for the average density of particles. And equation will be just the same. But the advantage of this method that we can take into account nonlinear reaction. So now look at this equation, governing equation for mean density of drug molecules. So you can see that the rate at which the antibiotic change depends on two terms. This is like transport operator, L rho, and minus, of course, degradation. And what we expect from our previous work that this degradation rate has a huge impact on transport operator on the right-hand side of that equation. So for local trap model, this operator has the following form. It looks like this. So now you can see that, of course, we have diffusion coefficient, depends on space, anomalous exponent here. But look at this term. You see, we have exponential factor involving integral with respect to time of this degradation rate. And of course, dependence of degradation rate on time depends not directly, but through the dependence of the density on time. So, of course, it's quite a complicated equation because the nonlinearity appears inside transport operator. But it's also advantage. And let me briefly, because I have only 15 minutes, describe the advantage to have this factor. So the rate, degradation rate, which depends on space and density, leads to the natural non-linear tempering of subdiffusion. So what does it mean? It means that in long time limits, we, instead of very complicated equations, we might have a classical diffusion equation. Because as I mentioned already, that if we introduce killing, then of course it kills those particles that trap for a long time. And so instead of power law, we have cutoff. But this cutoff, it's quite natural because of killing. So in a way, the killing process or degradation process encourage diffusion on average. And that's what happened if we consider long time limit of this equation. Then we got so-called reaction-enhanced diffusion. Let me explain that. So if we consider long time limit of that equation, then it will be classical diffusion equation because of cutoff, but with a very interesting result that I'm sure if you look at this equation, number 6, you recognize it's just a stationary solution of diffusion reaction equation, the classical one. But the difference is that diffusion coefficient now, d theta, depends on reaction rate. And also you see its anomalous exponents also there. So what I'm trying to say is that if we have that kind of large time asymptotic limit, then it's not simple diffusion reaction equation. Diffusion, because of this effect, depends on reaction. And this is pretty unusual because usually we consider diffusion is transport, it's nothing to do with reaction, and we have a reaction. But in this case, we have, you can see that diffusion, this is standard term. A is just the jump over 2 tau, tau naught is just specific time. So we have this classical expression for diffusion. But now we have this non-dimensional product of tau naught and nonlinear degradation rate. And we see also, it's very important, I mentioned already, this diffusion coefficient depends on anomalous exponent. Why this formula is important for us? Because the whole idea is to find the profile of antibiotics inside biofilms. That's the purpose of the research. Of course, we could do Monte Carlo simulation, agent-based models, but this is like can be measured in experiment, this profile. And I already mentioned that antibiotics fail to penetrate to biofilms, and we believe that this new equation can give us the idea why we have the failure of penetration of antibiotics in biofilms. So, but we don't know yet what will happen for barrier model because the equation is still not available. For that reason, in Manchester, in our group, we decided to work on that subject together with biophysicists and biologists. So we organized now a team of people, and they're going very soon to make experiments with the penetration of antibiotics into biofilms. And we hope to obtain the profile of antibiotics inside biofilms. Then we are going to derive also equation for barrier model. And then the idea is, based on experiments, we have to decide which model is better. The classical local trapping model, which is standard models nowadays in reaction-subdiffusion or variable order fractional PDE, or barrier model. Barrier model is not that popular in this fractional community, but it might be is better description of penetration of biofilms. We don't know yet. So finally, I have to mention research team. So it's Nikolai Korabel, my research assistant. We worked together for the last almost 10 years. My former PhD students, Daniel Han, now he's a lecturer in applied mathematics at the University of South Wales. And now we have a connection with Tom Way. He's a biophysicist. He wrote several books on biophysics, including biofilms, and he's going to make experiments for us and find real profile of antibiotics inside biofilms. And then we hope we will make a conclusion which model is better, barrier model or local model. Right. Thank you very much.
Thanks, Professor Fedotov, for the nice talk. For questions, please raise your hand. I'll call out your name in the order they appear on my screen. I don't see any raised hands, but if you still have a question, remark, or a comment, you can still unmute your mic and go ahead. I think okay. Uh, Please go ahead. I cannot pronounce your name. Shok. Yes.
Hello, Professor, and thank you very
Hello.
much for your interesting talk. My name is Shahzad Bek. Do you remember me?
Oh, yes, we met in Astana recently.
Yeah. Uh, what what do you think of uniqueness of the equations?
Oh,
Your
we, we,
models
we haven't
uniqueness.
done this yet. So at the moment, the problem is how to derive that equations. Of course, we did not analyze of existence, the uniqueness of the solutions. That's what I mentioned at the very beginning. Today, I focus on derivation of those equations rather than um, solutions of them. So that's my answer.
Mhm. Thank you very much, Professor.
I have a trivial question, if I may. So I missed the part. How did you derive the barrier model?
Which one? You mean?
It was much in the it was in the it was in the beginning. Uh,
Like this.
yes, this one's yes.
So that's local trap model. That's first derived by Chochkin Sokolov with Gorin flow. They use a symmetrical random walk.
Mm-hmm.
And of course, they use the heavy tail waiting time distribution. And they just use local model. Uh, But then I remember one of my colleague, Nepomniachtchi, asked me why we have this very strange diffusion equation, because in a classical diffusion setting, usually we have a term proportional to the gradient of P rather than gradient of the product. So, and then we managed to derive these new equations with a slight difference on microscopic level. But the solution of this equation is completely different.
I
So,
see. I see. So what
and
would
it's not clear which equation is right.
I see?
Both of them probably, depending on underlying random walk.
Mm-hmm. Mm-hmm. Okay, so my curiosity is like, how does one go ahead with it? For instance, I come from a bit continuum mechanics side, so then you're doing, mass balance, or like, some kind of a balance law or conservation law you have, and then you're deriving an equation. So, so deriving your equation, do you have some underlying strategy here?
Oh, yeah, of course. This is just the balance equation. Particle coming at the point, escaping. It's a very simple idea. But
Mm-hmm.
the question is, when the system is heterogeneous, where to evaluate heterogeneous rates? At barrier or inside box? So this is related to the famous problem like Ito-Stratanovich. So um, the difference between these two is we have extra, if you differentiate, for example, the first equation with respect to x, you've got two terms. One will be diffusional term and another will be drift.
Mm-hmm.
Here there is no drift.
Mm-hmm.
So that's a main. But here, as I mentioned, we have a very unusual anomalous drift involving I don't know. This is like looks for me like fractional operator. but the kernel, pretty unusual for me.
Okay. Okay.
But again, this is just technical things, but it's like chemotaxis.
Mm-hmm. Okay. I've not seen this such kind of no operator, so so I would look forward for your upcoming studies uh, uh, to read more about it. Okay. Are there any more questions or comments? You may unmute your mic. If there are none, I think this is a good point to close this talk in here. Thank you very much, Professor Fedotov, for the nice talk. We are very happy to have you here today.
Thank you very much. Thank you.
Thank you, Professor.